Then I'm really pleased to introduce Xerxes Arsiwalla, who is a member of the SPECS research group, which outstanding research group located here in Barcelona. And Xerxes is actually also a BCBT veteran. And he ended up with us via BCBT. He signed up for BCBT, and he was so inspired and impressed by the brain that he thought, look, I need to do more of that. And so he stayed here. Now, Xerxes doesn't like introductions, so that's why I make it a bit longer than usual. So in addition to being a well-accomplished athlete in extreme frisbee, he's also a highly accomplished mathematician, and that's exactly what Xerxes has been bringing together, coming to SPECS, to start to think more about brain and brain connectivity from a formal perspective. And I think it's going to be a fantastic presentation, Xerxes. I'm really happy that you're here, that you present this work, and I'm looking forward to what the brain does.
Thank you. Okay, so let's, should the lights off?
Lower the lights.
Okay, so let's first start by just being clear. What do we mean by complex networks? In principle, and what does complexity mean? Why are we interested in this question? So you can have a network as a lattice, or you can have a network as a regular graph. That's not a complex network. A complex network is usually defined as something whose topology is non-trivial, neither to regular nor to random. Complexity is in between regularity and randomness. Sometimes you might have heard of it being described as something that's between order and disorder. So biological networks, in fact, most of the time are in this class. And when we want to quantify complexity, what we want to quantify is the properties of these non-trivial topological interactions and the associated dynamics. We are not just looking at structure, but in biology we want to understand function. And to understand function, you look at how dynamics works on this kind of structure. And is there a way to quantify that? That's the question. Okay, so this is the overview of the talk. I mean, of course, you can always make this network bigger with other things, but this is just what I'm going to discuss on today. That's why it's a bit limited. We start with saying something about biological networks, the ones we are interested in, something about their features, what makes them special, how do we study them. Two main approaches that we'll follow. First is phenomenological. We want to model them, we want to understand local dynamics. And from that, try to play with the system, literally play with it, because you all know, and this was discussed few talks last week. When you do modeling, there is the problem of parameters. Most of the time, models are like that. You try to tune parameters to fit the data. So in a way, a model at best tries to explain some data at some level. I mean, the reason there are so many free parameters which you still don't know is because there might be underlying dynamics or mechanics beneath it. So besides modeling, obviously one needs to go ahead to try to know what's beneath that level. And so what we try to do over here in the lab is we also try to take a more principled approach towards brain systems. And among the principled approach, one is design principles. Paul mentioned a bit about that, which involves control engineering and brain architectures like the DAC. My talk won't be so much about this part. You heard about it in the previous talk. It will be more on another kind of organizing principles, which are global properties related to complexity measures, in this case based on information theory. And I will mention a bit about how they relate to consciousness, though this talk itself is not about consciousness, but it's more about how these measures can be used to understand certain principles and how you can apply it to clinical data like fMRI or some patient data. So that's the overview. So okay, let's just let me tell you what kind of biological networks potentially we are interested in. I'm just giving a kind of broad classification. At the highest level, we have the neuronal network. In fact, this is Hugman's data of DTI. So each of these nodes and links are actually not even neurons, but they are populations of neurons. Because if you go to a neuronal level, okay, this is another question. At what level should you study this brain system? And in a way, we are restricted by the data that we get. If your data is at a finer resolution, then you'll start modeling at the level to explain that data. But if you try to explain fMRI or DTI, then you will build models that are at the level of populations of neurons, not individual neurons. But we can talk about that later, okay? Then at a slightly lower level, they are the protein interaction networks, and even less, they are genetic regulatory networks. Now, actually, the protein interaction networks are quite complicated. I just chose this extremely simple version because maybe this might be relevant for a few talks later. There is John Lisman talking about, I'm guessing, about synapses. And
No, John will talk about basal ganglia hippocampus.
oh, okay. So this was his earlier work that he had worked on with synaptic plasticity, and this particular interaction network has to do with the CAM kinase molecule, and it was relevant for understanding why synapses can act as bistable switches. And then over here, there's another genetic network, not directly related to this, but this is actually what works as a genetic toggle switch. This one is not in humans, this one is in E. coli. But nonetheless, what you'll see in this talk are that all these networks can be modeled by stochastic dynamical system. In fact, this one is just a set of two differential equations which shows you switching behavior. This one is chemical kinetics, and when you go up to the level of the brain, the population models we describe, in fact, were obtained from earlier decision-making models which themselves showed switching behavior. So you will see there is some, though these levels of description are very different, there is some common principles among these networks. And we'll just try to, if you just try to make a mental checklist of what are these principles, so that once you at least have them in mind, then you know it guides you towards your modeling or towards defining measures. So one of the things about biological networks is that they are stochastic. There's noise, but they are also fluid. By fluid, I mean I just kept this in the same category because what I mean by fluidity really is non-specificity in a certain sense. So in an electrical network, you would have things connected by wires. In a cellular network or some other level of biological networking, you don't really have wires. You might have a diffusion gradients, and there is some chemical specificity. Not everything reacts with everything else, but there is some kind of statistical way of describing those interactions. So there are no real wires. There's no specificity. That's what I mean by the term fluidity. Maybe there's a better word for it. Stochasticity is noise. With noise also, it's not clear what noise is, whether noise is sometimes just noise or noise is because of underlying principles or an underlying description, a molecular mechanism or something beneath that higher level which we have not yet figured. Then there's plasticity. These networks are plastic. Now why I'm writing these down is because when we proceed with the modeling and the measures part, the complexity measures and all, we need to take into account these things. We cannot just build artificial models or artificial measures that don't care about this. So for instance, if you look at a basic textbook, they say the description of a neuron is often given as a RC circuit, a resistor capacitive circuit, which is a very simple circuit actually. You can put noise, but there's no noise in it, there's no plasticity, not much. And then the first time I heard of it, I was like, okay, if I just put like a few billion of them together on some machine, it should just do what the brain is doing, right? But it doesn't. And the reason it doesn't is because of the previous level slide I showed you, where you have several layers of description beneath the neuronal level. To put it the other way around, if neurons were just described at that level as a RC circuit, then we would be as slow as all our computers are in their simulation of the brain. So clearly there are these lower levels which make modeling the brain and all these large brain projects a bit questionable. I mean, it doesn't mean you shouldn't do it, but there's an approach to handle the problem, and here we are trying to lay out certain pros and cons in our own work related to it. So then there are developmental constraints. I won't have much to say about this today, but the theme of this school is on evolution and development, and you've heard quite a bit on that. And finally, this question of levels. It has been mentioned in a couple of talks, and towards the end we might discuss something about it, but again there is no answer on that as yet. So let's start with the first part, with the modeling, with the phenomenology. So what do we do here in specs? We are also, like many other groups, working on a big brain project, and like I said, the idea in these things is, let's say at the first approximation level, what you want to do is at least explain the data. So the data comes from DSI, DTI, and fMRI. So the game that people play is, you take structural connectivity, you sort of know something about neuronal dynamics from physiology and other studies. You build a dynamical model based impose that on your structural matrix and try to predict functional activity that you can compare with your actual fMRI signal. So if your model is good and your data is good enough, everything, you would expect that the predicted fMRI signals would match the real ones, and people try to get close. How good is not so clear at this stage, but at least this kind of approach might be useful when you compare between, say, patients and healthy control groups or something like that. So okay, over here, what we'll do over here is, one of the things is that in networks like the brain, which are rather, so in this case, the Hagmann's data which I showed you, which will come back here, this particular network consists of 999, almost 1000 nodes. Each node is a population of neurons. In this case, the population probably has like thousands of neurons. I don't have the exact number, and the connections are white matter axonal connections. In Henry Kennedy's talk and a few others, people have already criticized this kind of data by saying that this is not all. This is, in fact, Henry Kennedy gave a number. He said that what you see in white matter connectivity is just about 20% of all that there is. The rest is diffuse gray matter connectivity, which is not picked up by DTI and DSI, and you need other ways to tap that. Okay, but there might be computational approaches to add that information into this. I'll mention that, but first let's see what we can do just with this as it is. So what we do over here in the lab is, we have the experience induction machine. I think many of you are doing projects in it, so you have seen it. This is a mixed virtual reality chamber, and it has been used in the lab for quite a bit of previous work. In this case, what we are interested in is, we want to build a 3D reconstruction of the brain network, one that is interactive and dynamic. So usually you could do this stuff on a laptop, okay? It's not a problem, but the issue is that when the network is fairly complicated and you want to start playing with it, you want to start tuning parameters, you want to start fiddling with the topology, you don't have a clear intuition how to do it just by looking at a matrix on the screen, because then you don't have the intuition of what would guide you. There are quite a bit of parameters, and in the end the dynamics will look completely different. And indeed, so one of the approaches in handling big data that we take in the lab is, we try to combine computational power with human intuition. That's what the experience induction machine is trying to do. It's combining those two things by these kind of visualizations and dynamics. You'll hear more about this perhaps from your own projects and group leaders, where you can interact with this network. For this talk, the important thing is, what you can do is, you can start playing with this network, in the sense you can choose certain areas, you can disable nodes, you can activate nodes, you can change the representation of the network. You can do those kind of things in order to understand what's happening. So the main thing we want to know is, like I told you, these are dynamic networks. We want to understand how activity flows into it. If you activate one node, what all does it affect? This is like saying that if you get a visual stimulus, let's say in one of the lower visual areas of the brain, how far does it travel? And if you want to know that, and if you want to know its pathways in the brain for certain cognitive functions, then you try to model that in this kind of system. So in order to do this modeling, we need the right kind of dynamics, and that's what I'm going to describe here today. This is work, just to give credit where it is due. This is work done by with other collaborators from the lab, Ricardo, Alberto, Enrique, David, Pedro, Gustavo, who's a group leader in another lab in UPF, and Paul, of course. Most of these people are all in the audience, so feel free to talk to them later. And this is Alberto, actually. This also. Okay, so now, this is Hugman's data. In fact, this is how Hugman represented his network when he released his data. As you see, this kind of representation, just looking at a picture, might tell you something, but what you need is you need to know really what's going through these wires, what kind of signaling is happening, how much information is propagating, those kind of things. So okay, so the next thing is, on this structure we need to put dynamics. By dynamics I mean each node is a population of neurons. We know something about how neurons behave. Neurons are supposed to be some kind of RC circuits. So the question is, okay, how do populations of neurons behave? How can we model that? So luckily, this is not something you have to guess. You just have to look into the literature in physiology. There have been for years work on decision making. Paul mentioned a bit about the work of Encarni, who worked on these population models of decision making. In fact, that model was originally proposed by Wong and Wang in 2006. What you see here is a pool of neurons, two pools of neurons, external connections, local recurrent excitations, and inhibitions between them. And this model basically behaves like a bistable switch. So depending on initial conditions, the population that has a higher initial condition will go to an on state, the other to an off state, and vice versa. So it behaves like a switch. By switch I really mean literally that a switch has to choose between one or two, up and down. And according to when you try to, if you try to play with the switch, it won't just hang on in between, except if you're precisely there. A slight push will take it up or down. That's what this model does. And this is just modeled as two differential equations, first order. You can also put noise into this. And these models have been physiologically verified with monkey experiments, I believe in human also. This is work that Paul just described some time ago. Of course, he showed you slightly more complicated version of the model, but this is the basic version. So these models have been known and tested in decision making. People have known them. So what Gustavo Deco did was he extended these models into situations like this, where you need not 2 nodes, but you need like almost 1,000 or more. In his paper, of course, he didn't go all the way to 1,000. He went to 66 nodes, because that was how the classification in the atlas was. So he extended this to more nodes and tried to say, can we apply these physiologically verified models to predict whole brain activities? And basically this is the model that he used. I'm just what you need to know is that the reason I just wrote down this thing is to introduce the players of the game over here. So you have a population firing rate. So why population? So originally the one way to derive these models is you can start with a spiking neuron level, and then when you look at a spiking neuron level, you can perform an aggregation of the activity across a population, and when you aggregate it, you get these what is called as mean field models, which gives you the mean field activity of that population of spiking neurons. And in Gustavo's paper, he also verified that the simulations match if you actually do them for spiking neurons, which is computationally much heavy, or if you use this aggregate version. So he showed a match at this level. Okay, so there is some kind of direction there. It's not perfect, but at least it's going in some direction. And so you have a population firing rate. You have a synaptic gating variable, which is basically the fraction of open synaptic channels. In this case, for these kind of models, among the AMPA, GABA, and NMDA receptors, this only refers to the NMDA, because the NMDA has slow dynamics, whereas the AMPA, GABA are much faster, and so they ignore the dynamics of that. That's just to put the specifics in place. And then the other parameters are global coupling and synaptic coupling. Now, the point I want to make with this is, you don't need to dabble into every detail of this, but you might recognize that this set of dynamics is actually a generalization of the well-known Wilson-Cohen rate models. So, in the Wilson-Cohen rate models to describe neuronal populations, they would only talk about a firing rate, which would be like this guy. Here, in addition to a firing rate, you also have dynamics due to it's coming. You also have dynamics due to the synapses. The synaptic gating is not a constant or a steady state variable. It also evolves with time. So it's a generalization of those models, if you like to say, but see it that way. But of course, it's derived from spiking models, and these guys are precisely the same things that they used here for the decision making and now for the brain. So Gustavo tested this model with 66 nodes. Each node was denoting a region of the brain. So in Hugman's data, basically, if you divide it into 66 parts, each region will have subsets of nodes. What we now do is, since we have the technology in the XIM and the computational power, we wanted to go for the whole thing. So we wanted to then extend that model for the whole 998, and not just extend it for the heck of an incremental improvement, but to really try to play with the parameters and understand what you can do with it. Because after all, one thing is to check what parameters make sense, but the other thing is, even if you have parameters that are somehow fitting the data or matching the data, you want to perturb the dynamics to understand what this system is doing in terms of functions or dysfunctions. You want to lesion some nodes to see when you lesion something, how does the activity reorganize? How does the firing activity of the brain reorganize? And those kind of things, does the model match to the data? So those are the questions you want to ask. Okay, so
Xerxes, maybe to be a bit clear about this, you said, well, Ancarni's model was already proposed by Wong and Wang, right? But indeed, these rate models of decision making have a long tradition. But what Ancarni specifically changed to that was this idea of the monitoring system modulating competition and so on, right? Just to say it was not an identical model as that one.
Right.
Just for clarification.
Sure. Okay, thank you. Okay, so now let's put all this stuff and see what you get. So in the results I will show you, you will see what we have in the XIM as well as what you can do. Think of the XIM as a live three-dimensional data generator. You have a model, it generates data for you, it tells you what the system looks like, and then you can analyze that data as well. So the XIM right now is built in such a way, it's not just a visualization system or a simulation system alone. It also is interfaced with any analysis tool that you like. So right now it is interfaced with the MATLAB brain connectivity toolbox, which basically just does graph theory analysis, but for your specific purposes, you might customize it with other analysis tools. So I'll just give you something of what kind of results we have, not so much to care about the details. It's clearly not going to fit the data all the way, let me tell you that anyway. But you'll get a glimpse of how we try to play with these things and improve it to make some sense of what's going on in these networks. So first what we do is we just try to simulate the dynamics of the resting state. So using the dynamics, using the structural data, in the XIM what we did is, here what you have is, in all those models what happens is that the system starts, the simulation starts at some initial condition, and then the activity of the brain converges to some resting state. Resting state is basically a fixed point of the dynamical system. So after a point, each of these nodes attain some firing level. In this case, the color bar shows red is high firing, blue is low firing. This is, I'll just explain the thing in a minute. So this is the back view, top view, and the lateral views of the brain. The four guys just denote different levels of noise. So in these models, there's noise, but sure.
What units of noise measured?
In this case, it's just noise amplitude.
But what units, in what units, in the synaptic equation, you added, what units is that in?
Here it is. Okay.
So what units is the sigma in?
By units, you mean it's just an amplitude.
Okay, what is SI measured in?
What is what?
SI, DSI on top of the equation. What is SI measured?
Oh, it's SI is dimensionless. Yeah, SI is dimensionless. So yeah, so okay. So here I mean what I have is this is I've just taken 4 different noise amplitudes. The thing is that with noise usually we don't know how much to take. I mean with of course, different brains are different. The usual thing to do in these kind of modeling is just to average over brains, and you take some average value. Usually, when you look into literature, people don't really know how should you choose the amount of noise in the brain, if it is Gaussian noise at all, and how much. So we just said, okay, let's take a more agnostic approach. Let's just run this simulation with different levels of noise. So this is the standard, sigma denotes the standard deviation or the amplitude of the noise, and we just take it from a very low level to a reasonably high level until the network starts crashing. So what you notice over here is that, okay, so these are nodes that are quite active. The green ones are a little bit less, and the blue ones are relatively silent. You can see that across. In this case, what is more active and less active in a way depends on what kind of connections are coming to that region. Some regions are more heavily connected, others not so much. This data is averaged over subjects. For sure, if you take individual brains, you'll find all sorts of differences. Yes?
Are there inputs given to prime this, or is this just running that network, letting it run with starting from a random starting point?
So the input is actually there is an input current that's given into it, similar to like in these models, you see there is always an input current, I1, I2.
Well, yeah, in your Pinto model, in your Pinto version of the Wilson counting.
Yeah, so here the I0, that's the input.
So does that I0 have a form across the brain?
In this case, we just chose so I'll tell you how the value of I0 was chosen here. So what they did is, in the spiking neuron network that was done in Gustavo's paper, he just chose this I0 such that the results of the mean field model match the spiking neuron model. So the I0 and some more parameters were chosen by making sure that these two models more or less are saying the same thing. There wasn't a physiological basis to do that.
But can you go to the next slide? Isn't the parameter of where the components are going to be active is going to depend on the distribution of I0 as a function of your nodes?
The I0 was constant throughout.
Thank you.
Yeah, the same. In fact, it was just so over here the I0 was just taken a little bit above 0. The only reason they did that is because if you actually take 0 I0, which means no input, and this is relatively a simple dynamical system, the attractor will just go to the 0. So if you just take a non-zero I0, then the attractor is something non-trivial. And the other thing with the coupling parameter, what they found is that the way when they try to match the results of this model to data, they realize that the coupling seems to be sitting at the point where the network is just at the edge of a bifurcation. It's a nonlinear system, so there is a bifurcation, and it's just before that. And if you had 0 I0, you just get almost 0 activity, and that's what's going on. Okay, so so for now we took most of the parameters as they were in the earlier work, but we started playing with the noise, and I'll show you a few more surgical procedures with lesions and those things that we simulate. Now, what we notice immediately when we start tuning the noise, naively we would have thought that, okay, if you just push up the noise, things will just get hyperactive and chaotic, and everything will become, in a noisy way, it would become active in a weird way. But actually what happened is that when you started switching on the noise, the network just seemed to shut down. And you can go into details why that is happening, or even in this case, of course, as I said, this is just a parameter we are playing with, so we don't know how, we haven't yet matched it to data or anything. But one way of thinking of it might be that usually such a complicated system, its attractors are balancing each other in a very sort of dynamical way, and when you put noise, it might be just ruining the harmony or the synchrony of it. I mean, that's just, I'm just giving an intuitive description. You might look into details why noise is just ruining the attractor level. And what could be, so what I'm saying is that this kind of playing with stuff, you wouldn't just be able to do if you just looked at matrices and things. We got the idea after looking at the dynamics, we started looking at the patterns, and we started getting an idea of what stuff to tune. One thing that could be done is in future, we could compare with actual data of patients to see if really different levels of noise have something to do with aging. So for instance, if indeed it is the case that in data from people at older age, they show less activity, is it because of more noise or what? So this is just a suggestion from this kind of modeling. We still have to go forward and test it. So
Xerxes, are you saying that the resting state brain by necessity has to be, let's say, almost noise-free to operate?
it's not noise-free, but there is
minimal noise.
Minimal noise.
But your prediction then is that noise should be tightly controlled
Yes.
in a resting state network.
Yes.
So do you have any suggestion how that's done? Would you speculate like there's some modulator that is tuning signal-to-noise ratios or whatever?
If you look at engineering principles, yes, there are ways in which noise can be filtered. You can use feedback to control it. But then you have to look for how do you identify those feedback circuits over here? How do you identify those noise filters? When you just look at small systems, small networks, you actually build these kind of feedback gains and all that. Here one would have to identify that too.
Right, but can you say something about the level of noise that can be tolerated?
Yeah, I will show you something about that.
Oh, that's going to come.
Like in fact, so in this case, I'll just show you with these numbers at what point things crash. There's one or two slides later.
Okay.
So this is roughly saying the same thing. The only difference is that what we did is that in the network, we just extracted 3 different nodes as seeds, and in these seeds, this is the highest level of noise, lower, lower, lower. And in these seed nodes, we just try to extract the whole time series. As I said, this system, basically, you can think of it as a simulator or as a data generator. So in this case, we're just extracting the data and trying to see what the signals is. And then, I mean, typically, you can do your kind analysis with this signal or whatever. Here, I'm just trying to, we just try to see that, okay, at these seeds, when noise increases, what happens? So the same thing, as you start increasing the noise, the total activity goes down, and the seeds really then, towards the end, when noise is very high, are just plain noise. There is not much activity. Then over here, in a second shot, what we did is, more or less with the same level, we just we try to simulate a lesion. So what we did is, in areas in the parietal and the occipital lobe, now I don't remember the exact regions that we basically disabled the connections on a group of 52 nodes over here. So these 52 nodes, these are not all 52. You don't see all of them actually, because some of them are a little bit in the medial part. This is just a part of the view. So the dark ones are the disabled ones, and then with different levels of noise, we just wanted to see how much activity is still there. So of course, for the lower noise, the networks crashed much faster, and now we just want to see, okay, let's compare this signal, let's compare this activity to the resting state. Are there some inferences we can make on it? And if it's suggesting something. So, okay. So over here, we just try to plot some statistics on that. This figure over here is showing you for the resting state, the 4 levels of noise from the lowest to the highest. And this axis is actually showing you the total activity over all the nodes. We've not normalized it. Just the total activity over all the nodes in different levels of noise. So basically, over here, in this plot, what you have is the base denotes all the 1,000 nodes, and this is the mean firing rate at each node, which is steady in the resting state. and we just sum over all the mean firing activity over all the nodes, because we just want to compare how much the total activity differs across the resting state network when noise switches on. So at the first level you have a little less, then by the third level it comes quite a bit down, and towards the end it's almost crashing. And then now the interesting thing would be to compare it with the lesion. So how does the resting state network differ from the lesion network? We would expect that overall in the lesion network activity patterns would be affected. So exactly you can quantify that, and this is just a crude kind of setup to see some basics. You could go more into details with the signals. You could try to see if somehow pathways have reorganized or something. I mean, there are more things you could do with it, but right now, before trying to do more things, one would have to really understand how close are we to certain experimental setups, what kind of parameter space should we be operating in to actually compare with real data. So those are things that yet have to be done. What this graph is showing you is for all of these nodes over here, we've just taken the difference of the activity at each node for the same level of noise for the resting state minus the focal lesion state, and the gray areas are the actual lesion areas. That's why there's a high spike. But the other places are where there was no lesion, but anything on the positive side denotes the amount of excess activity in the resting state compared to the lesion state on that node. Now, as you increase noise, what happens is you could just think that one thing we might have thought is, okay, with noise, sure, the activity goes down, but the difference might be the same. But what actually turns out is the difference isn't the same. By the time you reach the third level of noise, the difference significantly increases from the top one, and that's also shown here. The first two levels are not so different, but by the time you reach this threshold, the lesion network seems to break down much faster. And of course, that might have some dynamical reasons. We don't know in detail in why that might be happening. But it seems that a noisier network seems to be less resilient to such a lesion. And indeed, if a noisier network has something to do with a more aged network, it might just suggest that one could look into such data to see if lesions at different noise levels or different ages.
But this should relate to the damping that occurs in your network, right? And the damping would come from your inhibitory interactions in your network.
In this case, what happens is that each of these, so the way the modeling is done is, right now there are no inhibitory interactions explicitly put. What happens is each node is modeled as a population that has 20% inhibitory and 80% excitatory.
Yeah.
But across nodes, they are only excitatory connections.
Exactly right.
Yeah.
Exactly right. So the point then is that you must be able to relate this damping factor, which only happens within the nodes, to the noise level at which the network breaks down. Right? So can you give us an intuition about that?
Here, that is not there. Okay. So what could be done is
you take 5% of your nodes, you take out.
Yeah.
Right? And if you take out 5% of your nodes, then with this noise level of 0.7 or something, whatever the value was, at the third level of noise, the network destabilizes, right? So that means the damping factor that you have removed is now overcome by this added noise. So there must be a relationship between damping and noise. So I was wondering whether you understand that relationship, how that scales.
No, I don't know how I would extract that from the model here.
All right. No, don't worry. It was intuition on that.
Yeah, sure. But I mean, so whatever inhibition is happening is within this, is somehow encoded within this nonlinear function. Right. And if you look at the properties of this function across its parameters, then you might try to say, look where this inhibition is really hitting when combined with the noise. So that might be something to play with at this stage. The other approach is to sort of try slightly improve this model by adding another line where you don't just treat a population as one, but each population itself has an excitatory and an inhibitory population combined together. So that could be a slight improvement in the model, and perhaps that might that might make it easier to see this damping than right now over here. So that's another possibility. So the stuff that we have been doing here is at this stage quite it's just phenomenological. We are playing with parameters. We still have to match it with data, but at least we are trying to look at the landscape of possible solutions that the model gives. Because what usually people do is they just tune all the parameters in a way to match data. So what you can do is now the signals that you have had there, for instance, at each node you have these signals. So what people usually do is, this is neural activity. They then pass it through a balloon Winkessel model, which is a kind of low pass filter, and that filtering is what is mimicking an fMRI model, and what comes out is believed to be an fMRI signal, which is a slow pass version of this. So then people take that fMRI signal, they build a correlation matrix and try to compare the correlation matrix of the model with the real data. And in order to compare it, you tune the couplings and certain things so that you get a best fit. So that's how it is usually done. And for sure, if you make improvements in the dynamics of the model in the details, you have to start pushing these couplings all over again. So that's the problem. Over here, finally, what we did was just to get an idea of where the lesion is affecting the network. Now, one thing is to do graphs like this if you know what to look for. But when you are not sure what to look for, you need something that you can actually play with. So what we did over here is we just took all these nodes that were lesioned previously, and we mapped their efference. So all the places that they project out to, and then there's a whole bunch of regions, part in the frontal brain, in the middle part of it. So we just wanted to know what happens at those nodes. So another approach could be that, all right, in the case of the stroke, we just cut off the connections. But now, instead of cutting off the connections, let's do something that is like a TMS, where we just shut down all the connections, we just inhibit all the connections. And if you inhibit these nodes, what happens? You'd expect that most of the network is inhibited, which indeed was the case. Like this axis shows the difference between the resting state and the inhibited network, and anything on top of the 0 is showing you an inhibition as a result of this TMSing. But what you also see is that, and the gray shaded area denotes the nodes, the site that was inhibited. Okay, so we don't put that activity because that is extremely inhibited. So, but what you notice is that when you apply such an inhibition to this whole region, it's not just that everything shuts down. A few things are actually excited compared to the resting state. There's not too much to make of it at this stage. So, for example, this is a population of neurons in the fusiform gyrus. This seems to get excited more than the resting state when it's inhibited. At the moment, it doesn't mean too much. What's probably happening is the following, that this region that is excited is, in the normal case, it's probably anti-correlated to the site that was inhibited or lesion. And because of the anti-correlation, now that this is shut down, this has just switched off, has just switched on. So there are these anti-correlations in the brain. We know about them. They are much weaker or much fewer, let's say, than the correlations. But what can be interesting about these anti-correlations is that when you look at patient data like stroke, coma, or something, most of the time there is not much activity. Things are just shut down. But if you actually study the model, what these kind of models are telling you is that they might be, depending on the type of lesion, specific areas might actually be quite active. So when you're trying to look at, for example, people in coma and trying to determine that what's the state of their consciousness, then you might, based on these models, it might actually guide you where to look for. So that's what I meant by this approach we are taking to phenomenology. For certain, whatever I've shown you is, at this stage, the work we have done is just a kind of proof of principle. We still have to, we build a system, we based it on as realistic dynamics as we could get, and we've tried to obtain some qualitative features. The next stage would be for sure to match it with data. We have yet to do that, but today this is what I wanted to show. Now, one of the things, the reason I'm showing you this stuff, which is to sort of drive the point that, all right, At one level, the way to analyze these networks is these kind of local perturbations, where you perturb parts of the thing and try to look at emergent activity pathways, if there is any kind of cognitive way to relate it to experiments, etc. But another thing to do is you can ask about principles of or mechanisms behind these dynamics, and part of it is what Paul alluded to. You could use ideas from control engineering to ask, when there is a modulation, what are the feedback loops that are doing that, or things like that. In this talk, what we are interested in doing is a slightly different question. So, yeah, so the perturbation analysis is what you saw, and the other thing, of course, you can do is you can just do network statistics and look at its spectrum and those things. But let's just look at this issue of synchrony, because this was the reason I'm talking about this is because you already heard this in Murray Shanahan's talk. In his networks, he told you, he showed that under certain conditions, dynamical systems show synchrony, and the way he tried to interpret it is that when two populations, not everything synchronizes at the same time, but when two populations synchronize, it probably indicates an exchange of information between them or some information flow. So if we want to make this idea a little bit more clear, also if you remember, what he said is that when a network is most of the time, you don't synchrony doesn't mean that everything just fires at the same time, and this is a little bit to do with the complexity of these interactions. It doesn't mean that everything is active. So just think of this as a kind of an orchestra. You don't have all the notes beating at the same time. To make music, you need a modulation of a kind of combination of things, some high, some low. You don't have everything shut or everything off. So that's in a way between complete order or complete disorder, and often complexity is qualitatively described as being a state between the two. So let's try to ask this question a little bit more quantitatively. How can we really quantify this notion of a whole being greater than the sum of its parts? How can in networks, in specific case of these dynamical networks, how can we do that? And related to that concept is that complex networks are globally integrated and locally differentiated, which means that on the whole there is a global. Globally they are well connected, but locally the parts are doing some different things. So these two notions, can we somehow quantify them? And one way to quantify them would be to go into the language of information theory. So the reason information theory is interesting is because, so for instance, if it was just simple networks that you come across, like not simple, but relatively simple lattice models that you see in physics, like the Ising model or something, which are usually equilibrium systems, you might just talk about energy states. But when something is not exactly an equilibrium system, it's a dynamic system and time matters, then you can't just talk about, it's not easy to talk about just stationary energy states. Then you need to talk about some kind of transitions, some flow. And in this case, from whatever we have learned in methods of communication theory and information, information is a good way to quantify some of this flow. In fact, using this language, we will realize that not only can we describe these kind of neuronal systems, but you might use a similar language for other systems like decision-making systems or even a genetic switch. So some of these methods will apply to some of the other figures that you saw earlier. So let's just to put the definitions on place, let's talk about just introduce the players of the game. So what we have over here is first, so in this case, let's say the networks we care about are stochastic and they are dynamic. In fact, they'll also be plastic, but I'll talk about plasticity later. They are just dynamic, stochastic, and plastic. As yet, I'm not even worrying about the multi-level structure, which is not something I'll have much to say on. So when you have a network like this, you can think of the random variable as a vector of all the nodes, like in the networks you saw. Just take a vector of all the nodes, and each node is a random variable having some value, some mean value, and some variance of that. So you can for such a system, or even if in this case I'm just putting the definition in a simple way, for an individual random variable, you can talk about something called an entropy, which is defined by in terms of its probability distribution. So this event takes values from a distribution. In this case, let's take the more practical example rather than making things too formal. Most of the time what we'll be doing is the variable will denote the firing rate of the nodes, and the distributions will just be Gaussian. So around some mean firing, some noise. So it will mostly just be Gaussian distributions, and you can compute an entropy like that. And entropy basically intuitively is just telling you about, it's a measure of uncertainty. So one way to think about it is that when entropy is 0 or extremely small, the system is considered to be in complete order. There's almost not much variation in the values that the variable can take. Whereas when the entropy is very large, you can think of the system as being in complete disorder, and every variable within that system can fluctuate wildly. So the idea is that it's not just that okay, and the way information relates to entropy is more or less can be thought like this, that the less likely an event, the more informative it is when it occurs. So in that sense, people often just talk about the entropy as an average the average of information. So precisely, I mean, people define information as minus log p. So entropy is the average information. Sometimes they just use these terms together. They just, when they talk about information, they are most of the time just referring to entropy. So in these cases, in many of these systems, as you saw, which are stochastic, dynamic, and nonlinear even, it's not always easy to do this kind of Hopfield type or Ising model type analysis. You might run into, you might not be able to solve it analytically, and you might have a tough time even finding its states and everything. So instead, talking about information sometimes avoids you the problem of trying to solve the whole system or the partition function, if it means something, and just talk in terms of information flow and try to understand. Tell me.
One more interesting thing about entropy, maybe for people who haven't worked with it
Sure,
before, is
sure.
additive. So you can describe the loss of information or gain of information.
That's exactly what we'll go into. Yeah, no, but go on. Finish your point.
Well, that was it, really.
Okay.
That's another useful.
Sure.
I think everyone's heard. That's okay.
All right. So as I said, as I said, it's so though entropy goes with information, the question is not about less information or more information. The question is about transitions or gains, and that's what matters. In many of these systems, you make jumps from one state to another. What happens that time? So that's what we want to quantify, and in a way, that's related to this question we had earlier: that is the whole greater than the sum of its parts, and how much? So these kind of things actually tell you how. So if the parts were just operating independently, that would be like, you know, each thing is doing its own thing, and that's almost like some kind of noise. Whereas if things operate in synchrony, then there is music. I'm just making the analogy. And here, when there is some such synchrony, which we'll actually try to compute by looking at the eigenmodes later, how can you quantify that in terms of the information gain? So that's how we want to now, we're trying to formulate the question, make it more precise in that sense. So when we started looking for ways to define such a measure, the first thing that came across in the literature was this Tononi's measure of consciousness. So such a measure phi, talking about information gain or whole versus sum of the parts, was already discussed. Actually, even before Tononi, actually it was Tononi only, but in several earlier versions, Tononi, Edelman, Spohns, they had already discussed things like neural complexity and integrated information. And right now, actually, this field within if you go to some conferences on consciousness or computational consciousness, there's quite a bit of talk on these kind of information theoretic measures on defining, they call it defining, but you don't have to take the strictest version of the way they propose it. So basically Tononi is trying to say is that the kind of networks Tononi works with, he's just using Boolean networks, that's more like a spiking neuron on off, but not with the detailed spiking dynamics. And in those networks he's formulating this question of how much does the network do as a whole more than the sum of its parts. And I'll write down the definition in a slide later, but now I'm just trying to tell you what he's doing. So basically the network makes a transition from an initial state to a final state. When it makes this transition, how much information does it gain as a whole compared to the sum of its parts? So okay, so why? So this is one way to frame it. What's interesting in this concept is that this notion is a bit related to something we have already seen in physiology, which is small world networks, which basically are also in fMRI and other kinds of connectivity data. People have looked at brain networks and tried to identify some structures which seem to be as if there is a global integration, but locally things are doing very different things. They are locally differentiated. And one of the things was in many of these works that people they do on consciousness, they try to say that the more of this is somehow correlated to consciousness. In fact, some people go so strict to say that this integrated information is consciousness. I don't understand that in that version, but the approach I take is that I don't care so much about defining a notion of consciousness. What I would rather like to do is, if indeed there is this way of looking at dynamical systems in terms of its global information or in terms of its emergent information, can we use that as a comparative tool between patient and control, for instance, or to know about mechanisms when you break down certain parts of the network, what's actually happening as a dynamical system, what kind of music is shutting down and what kind of music is still on. So it might be more interesting to look at it that way, and maybe for sure later if you look at actual data of people in different levels of consciousness, clinically you might use these measures for that, and then you can say that there is some correlate to information processing and levels of consciousness. But I'm just stopping short of making this correlation into some sort of philosophical identity about the nature of consciousness. So okay, but in any case, the way Tononi defines his measure, it's sort of useless for the purpose we have here, because not only is he dealing with just Boolean networks. So in this case we have networks that are having continuous time series data. So now we have to formulate this version for time series data. And the other problem with Tononi is that the way he defines his measure, it is impossible to do any computation for networks that are more than like 5-10 nodes, because he defines a way of partitioning which then makes it computationally intractable. The way of partitioning he defines itself we don't actually agree with. We means the work we are doing here, Paul and me. So we try to take another kind of partitioning of the network to talk about what it means to be the parts and what it means to be the whole. And in terms of that, we actually solve this problem of this computational intractability, and we also try to interpret what it might be doing, slightly different from the version he's proposing, and then we compare our measure to the actual dynamics of the network. So it's one thing to say, okay, I have an information measure and it's telling me this much information gained from year to year. But when that happened, what did the system do? How can we look at the states of the system and actually see that the dynamical eigenmodes of the system confer to that measure that we have computed? So okay, so you don't have to worry too much about the mathematical formulation, but the way people in information theory formulate a question of information gain is by what is called a Kullback-Leibniz divergence, which is comparing two distributions, one a distribution of the whole and another a joint distribution of the parts. So there are ways in statistics to just compare two distributions, and here when you make such a comparison and then you just do some algebra, I'll just give you the end result, The end result is relatively practical. The end result for phi, the measure, is just based on a quantity which is sigma, which is actually the covariance matrix. In this case, the conditional covariance matrix. So basically what phi is asking is that when a network makes a transition from an initial state to some final state, this is the covariance of the initial state given that you observe the final state. So basically, observing the final state, how much information do you gain about the initial state when you observe the final state and subtract what is happening from the paths. So this was the whole versus paths thing, and that's encoded in that definition of comparing two distributions of the whole and the paths. And so what's over here, this is stuff I already explained, but now that you know what it means intuitively, it will not look too formal at this stage. So this is the initial state of the network. These would be in this case the 1000 nodes or whatever. It goes from some initial state to some final state. The distributions are Gaussian, as I told you. So there is a mean level, and then there is some variance at each node. And in the results I'm showing you here, I've just taken a simple generative model. The dynamics is just linear dynamics. So this is like Markovian dynamics. You have an initial vector, a final vector. This is a connectivity matrix. So this would be the actual structural data and some noise. And this work at this level, also Anil Seth and Adam Barrett have worked on this version of the measure. But again, theirs is slightly different. They more or less are sticking quite close to Tononi's way of partitioning a network, and they also get stuck with the same problem of not being able to extend it computationally. So what I'll come back to that slide. But what we do over here is we choose a different way of partitioning the network, and what happens is that here I'm just giving you a comparison. These are the number of nodes. You can start to, okay, this is trivial. 1 is trivial. You can have a network of 10 nodes, 100 nodes, and so on. If you do the way Tononi and others do the partitioning, you have to consider these many configurations, which after a point become too much. Whereas if you do the way we are partitioning, I'll tell you what it is. There's just a unique way of doing it. And not only, it's not just about, okay, being unique means, sure, that means it's computationally makes your life easier. But I'll also show you that there's a sense in doing it when you actually look at the dynamics of the system. So what is this partitioning we are talking about? So a partitioning of the network is basically trying to say what are the paths. Now what was done up to now was that what people did is for the paths, they just divided the network into two paths. Here what we are saying is instead of talking about two paths, really talk about the paths, each node as a path, as a part of the network. So divided into all the paths. One of the problems when you try to coarse grain the network like they were doing into parts that themselves are composed of subparts, there might be some mathematical subtleties there. When you try to coarse grain systems, you might have to check if the dynamics that you thought works on the level of the nodes also works on the level of a coarse grain partition of the system. There are people who are working on what is called effective dynamics, which is sometimes telling you that when you take a subset of a network, the dynamics is not just what is a collective thing of all the nodes. There is a way to effectively average the dynamics when you're considering paths as two paths or a subpart like that. Whereas when you just do it with every individual node, then at least we are doing the right thing by definition. So that is one thing, but later you also see some results to support that thing. So the partition we use over here is basically, instead of what Tononi calls the minimum information partition, what we use is the maximum information partition. Because when you part a network and you keep parting till, yes?
So you still have a decomposition of the system into nodes, except that you're basing yours on the data.
Yes.
So it depends whatever was the granularity of the measurement of the data that you're taking.
Yes.
So you still you don't get away from that problem.
No, I don't. But I'll tell you what. So what I do is, at this level, I want to just address this question for the data I have, and then you can start speculating if you have more structure, what goes beyond and how much it can add to. So at least I will just try to. That's definitely the point. I mean, I'm not trying to escape from that, but I'll just put it on later. And at some level, I'll not even have an answer for it. So, okay, So basically what I told you, all right. So in this kind of, so what we have is, we have initial state, final state. I'm just showing you one jump. You can then extend these methods for multiple states or things like that. I have simple linear dynamics. The entropy of a Gaussian distribution is known to depend on the covariance matrix. And the measure has a relatively simple formulation, but well, this is little, it looks less less simple than it actually is, in the sense that this covariance matrix, one thing you can do is just go numerically and put in numbers, but when you put in numbers, sometimes you don't know when something is happening weird, it's whether it's an artifact or something to do with the dynamics. So instead, for the smaller networks, all the results I show you are analytically computed. There is a given a structural connectivity and white noise, you can actually compute the covariance matrix explicitly and analytically, what I mean, and also the partial covariance matrix. I mean, just for those who are a little bit into it, what will happen is that from this, I'm not showing it here, but you get the discrete Lyapunov equation, which actually you can solve for the covariance matrix of the signal, and then from that there's an identity that will bring you to the partial covariances as well. But the calculations can be a little bit involved with the algebra, doesn't matter. Point is that you can, for relatively simple networks, you can get an explicit result. And what I'll do is, I'll choose my networks with plasticity, so the results we get, you can tune it over the synaptic strengths or the edge strengths, whatever you would like to call it.
But now, Xerxes, I could argue that, so you started with this definition of Tononi, of, let's say, in his case, a conscious state, but you could actually look at brain dynamics in general, right, where you say, look, information of the whole should be more than that of the parts, right? So it's a general issue. But I could argue you've sort of changed the definition, because in the Tononi case, you have to really look at all possible partitions of your network, of all possible subgroupings of nodes, and then you have to do a specific comparison, like is the whole more than the parts, right? But you seem to have redefined this because you don't have to worry anymore about this combinatorial explosion. So I could argue, well, actually, you have just defined a new measure. You have not improved the measure, but you have just changed the measure qualitatively. Would you agree with that? So have you qualitatively changed the
right.
measure, or just
So the way I would say it's a change, but whether the change is a big change, a small change, I don't know how to say that. I don't have a measure to say that. But the interpretation is the following: that let's say you take a network and split it into two parts, you get some value of phi. You split it into two more parts, you get a higher value of phi, and you go on till the last. So when you split it into all its parts, the phi is the maximum, and that you can show exactly. It really is the maximum of all the phis. So what we are having is not the minimum information that you can do by dividing two parts, but the maximum. And the maximum, later you'll see, is more robust when you start playing with the network weights and plasticity and those kind of properties. So that's the difference in how I would interpret it. And in a way, this interpretation makes more sense, because it's, if you really care about how much it's doing as a whole, then you want to know all of what it's doing as a whole compared to every part, not just compared to a subgroup. And if you want to do it compared to a subgroup, maybe it would be more precise to actually define the average, the effective dynamics of those subgroups, which people have not usually thought about it that way. Yes.
I'm very pleased you chose to sit so far in the back.
You started with the Wilson-Cowan equations, and now this information measure is all based on linear equations. Is that, I guess I have two questions. One, is that because you can't actually analyze the Wilson-Cowan equations in this kind of complex information state, and or is there a reason for doing linear? And two, wouldn't actually having the nonlinearities change all of this completely?
Okay. So the way you can analyze the nonlinear is, the reason is linear here is because I wanted to get analytic results. If I would put it at nonlinear, I might not be able to solve all the equations explicitly. I might need to just get numerical solutions, which is fine. but here it was more for principle. So the way you do it, if you have, so as you notice also, this was a discrete time linear equation. So if you have a Wilson-Cohen or even like the mean field models that we actually use in the brain model, what you can do is, first you can just discretize the system in the typical Euler discretization, which is anyway the way people solve it. So you discretize it, and then you basically get it, you get it in terms of, so let me just, so when you do the discretization, what you'll get over here is not just a simple connectivity matrix, but you'll get a function of that. And then, well, so once you get a function of that, you might still try to, given that nonlinear equation, you might still try to take the covariance on both sides and get the nonlinear discrete Lyapunov equations, but then you need to solve it numerically. And then you get all the values, all the entries of the covariance matrix, but as numbers, depending on the couplings and other parameters. And once you get those numbers, this still holds, because this is, okay, so if the model is still Gaussian, this still holds. If the model is not Gaussian and you want yet another level of precision there, what you could do is, you have the system, then you could try to determine the actual probability distribution by solving the Fokker-Planck equations for the dynamical system, and then put that in. So you could actually do all of that. Of course, if it's Gaussian, life is just much easier, but numerically then you put stuff in and get it here. And you can continue, so what holds if you do all that? The way I partition has nothing to do with the nonlinearity, so that still is valid. If it's Gaussian, this equation is still valid, and the only difference is that then you put in numbers, so most of it holds. I mean, not something that comes at the top of my mind that completely differs, but of course, what will drastically change will be the actual dynamics, and the dynamics I show you. So right now I'm just solving this for stationary dynamics. When you have a nonlinear model, things get more interesting because there's not one attractor in a nonlinear model, like in this case there's just one attractor. There you have multiple attractors and you might have bifurcations and all that. So things might get more interesting, I would say, not worse, so to say. And that would be the interesting thing to, if we actually want to start using these measures for real brain data or even for these monkey data of decision models. That's what one would need to do. Okay, so let's just apply this to the linear model. I'll just show you some prototypical networks. Now let me just describe what I have quickly. All of these networks have 10 nodes. These links have a connection strength G. All of them at the moment have the same strength. You can just play with them and do other things with it, but I just want to get something that could be sort of tractable. So I put all the connection strengths as the same, the coupling G, and I just take different topologies, and there's another slide with some more. And then what I do is I just start computing the phi. In this case, I'm not showing the actual function of this. They are just functions of the coupling strength G, which are exactly determined, and I'm plotting them here because if you just look at the actual equation, it might not mean much. So if I just plot the phis for this and just take an overview first, and there are some more networks. Basically, what I've done is this is a completely uniform network. Everything is connected to everything else. This is just cyclic. This is more like local connections and few long-range connections. This is again quite regular. This is a little bit random. And here I just purposely chose this example of a network that is not bipartite just to see it, but it didn't. The results were still okay, so I didn't have to care much about it. So what you see is that, all right, you have a formula for phi. You have a network that that has different connections, some dynamics, and what we found is that on this axis we have the coupling strength G, which is the same coupling for all the networks. So you can tune this coupling from 0, which means nothing connected, to something very strong, and then you can compute phi. And then what happens is the profile of the function of phi shows you these kind of peaks or resonances. Now normally people did not discuss this with noise, with plasticity and everything. So if you consider plasticity, which means these strengths change, what happens is that across the different synaptic strengths, the profile of how much information is generated or gained as a whole changes. So I'll just give you a little bit of the interpretation of the dynamics of these peaks in phi and if this phi is really the measure of complexity, it seems like there are certain levels, points at which complexity really peaks. What are these? And when it happens, what's happening in the network? That's what you're interested in. Is there really something interesting and non-trivial? So, okay. So, first thing, just to be clear about so the solutions over here that I'm giving are only for stationary dynamics. You can extend them to non-stationary or metastable states, but right now I'm just doing it for a stationary system, so forget about metastable states at the moment. And what you'll realize is if you just look at, since this is a nice, well-defined, clear, simple, linear, analytic, dynamical system, you can try to look where it is stable and where it is not stable. The solutions are not stable for any coupling. So in fact, in all these cases, for stationary dynamics, what you should look at is until the first peak, until the first pole, the dynamics is stable and stationary. After that, stationary dynamics is not stable. You might look for stable, non-stationary or metastable solutions, but they are not stationary at least. Nonetheless, what these resonances are, we'll mention a bit about them. So anyway, if we just look at the stationary regime, which means until the first point, it seems like at some point for all of these networks, the measure of complexity or according to this measure, complexity seems to be peaking into something very high. Then the network becomes unstable or critical, if you want to say, and then the solution stops until that point. So, which seems to suggest that that complexity seems to be increasing when you're at the edge of a when you're at the edge of stability, and what we want to understand is what this means in terms of the synchrony that I alluded to much earlier when we were trying to ask about what it means. So let's address that. So first thing would be that, okay, we have since these are dynamical networks, just think of each node as generating a time series, okay? Each node has a time series, and in this time series, like a fMRI experiment, if this was the brain and you had your time series, you take all the time series data, and in that time series, all these signals, you can do statistics with it. You can look at the correlation matrix of that time series, or you can look at the principal eigenmodes of the time series. So basically, the principal eigenmodes will tell you will basically tell you how to explain the data in terms of some PCA or something like that. So if you look at the covariance matrix, I've just taken two specific networks, the third and the last one, against the global coupling. If you look at the eigenvalues of the covariance matrix, which is the covariance matrix is the matrix of covariances of the signals that are coming from the time series of that network, its eigenvalues will basically tell you the principal components of that signal, which means that for, like they say in statistics, that a large eigenvalue for a given eigenmode means most of the data can be explained just using that mode. This is the idea of PCA or other related algorithms. So here what happens is that if you look at the eigenvalues of this matrix, it's a 10-dimensional network, so there will be 10 eigenvalues denoting 10 different eigenmodes. And what are these eigenmodes basically? Intuitively, these eigenmodes are so a trivial way to think of a network is to think of each node, but an eigenmode is not a single node. It's all the nodes at different values of strength. The strength could be, in this case, the strength of the signal. And so basically an eigenmode is like, if you think of the network as a drum or something, and the nodes as giving a signal, the different shapes of the drum are the eigenmodes, and if you choose them in the right way, a linear superposition of these modes generate the whole signal. Quite similar, analogous in intuition to when you think of Fourier decomposition or something. So basically, the eigenmodes of a dynamical system can be linearly superposed to reconstruct the whole signal of the system. So what happens over here is that if you look at the nature of these eigenmodes, the eigenvalues, you realize that they also are a function of plasticity, and it seems like exactly at those points when phi seem to hit a very high value or a resonance, the specific eigenvalues of certain eigenmodes also seem to peak. And then as you go to another value, so what happens is that as you start, think of this as a game where you're just trying to switch on the coupling from something very weak to very strong. As you start tuning on the coupling, some eigenmode is dominant, and the vibrations of the network are guided by that dominant eigenmode, and that dominant eigenmode becomes more and more dominant, and high phi seems to correlate to the dominance of that eigenmode. And this eigenmode is exactly the idea of synchrony that Murray Shanahan and others were speaking about, in the sense that certain populations are synchronized and others are at a low value. And so here what you see is that high synchrony is associated to a high information gain. So indeed this is a quantification of that notion that we saw some time ago. And here it is not just a gain of information, but it is how much is more beyond after subtracting what the paths do. So really like a collective dynamics of the thing. So basically what you notice over here is in this analysis is that you tune the coupling, an eigenmode dominates, then the network becomes unstable, the eigenmode falls down or becomes weak, and as you tune on, at some other point another eigenmode will then become dominant in the non-stationary regime. So it's almost like that certain eigenmodes are building up, and there's high synchrony and there's high complexity, lots of information gained in that system, and at some point there's an information overload and it breaks down and the system becomes unstable. And then again it tries to the system tries to build up as you go to finds another configuration where another eigenmode or another mode of vibration dominates, and information is exchanged among those among the subparts of that eigenmode, and there's a lot of information exchange and high complexity. So here basically we have associated the meaning of this complexity measure to the specific eigenmodes. So it sort of relates to the intuition of what happens when parts of the network synchronize. Not everything, phi doesn't go up when everything synchronizes or when everything shuts down. It goes up when there's a combination of things, exactly like how you would have harmony in a musical orchestra. You can't play everything at the same time or nothing. You need some kind of balance, and in a way this is putting that notion to test in terms of information gain. And one thing what we can do is, all right, this slide is now not so important given the time, but the point over here was that we just made a comparison of our measure with other existing measures, and this was the point I said. According to how we do the partitioning, the results change, and what happens is the red line. So what I did over here is, this is a network of 10 nodes. If you do the bipartitioning of this network, you can split a network of 10 nodes into 2 equal parts in 126 ways. It's there, okay, I'm not cooking up the number. And so for each of these 126 configurations, in this case, I explicitly compute the phi analytically, and these green lines are all that, and the red line is what we have. Now what you see over here is, then here's just a zoom up of that. So phi, the way it is defined as a Kullback-Leibniz divergence, should always be positive. It has some properties, and what you see is that the way we've defined a measure, and this is the point where of stationarity breaks down. So until the stationary limit is reached, our measure smoothly goes up, makes sense, and relates to what we think the eigenmodes are doing. So the measure we define seems to tally with what the eigenmodes are doing. Whereas if you look at the other versions, you see that they actually are, first of all, not robust against changes in plasticity. They erratically sometimes even become negative, which means they are not even defined in certain parts of the stationary solution space. And there is no notion of anything being unique. What starts out as a maximum or a minimum fluctuates, whereas this is uniquely the maximum at all parts of the solution space where the stationary solution holds. And it is robust under plasticity, and it correlates to what the eigenmodes are doing. It doesn't have certain, it doesn't break down in a way that would indicate that something is wrong with the definition. So like I said, what must be wrong is that when you do the paths, you have to be careful what is the effective dynamics of those paths. That would be my take on it. But anyway, what we see here is there is a nice way to define this measure and to make sense of it, and then you interpret it as the maximum information gained by the whole over the sum of every part, and so the next thing would be to
But Xerxes, wait.
yes.
You could now argue that this is all fine for a restricted range of coupling, because at the high coupling strength, then your measure seems to underperform compared to the specific test cases you looked at.
It's
Right? You say it uniquely captures the maximum, but that's only true at the left-hand side of the graph. At the higher levels of coupling, there you seem to underperform compared to these test cases you looked at.
so at those levels, you don't apply these solutions, because here I explicitly solved for a stationary regime. What you do is you really consider a non-stationary regime. So one, I just tried it out. I don't have results here. What you can try out is you can start these networks. You don't do anything; they're just there, so to say. You can start putting external driving forces, and you can start looking at not just one transition but a few transitions where the network jumps, and then you can you have to then just recompute the function at that regime and do it to make sense. So right now, all that these plots tell you is that even in the non-stationary regime, certain points where the poles hit, those poles will be the same because they have something to do with the spectrum of the network, which doesn't matter whether it's stationary or non-stationary. So if you do things correctly in the non-stationary regime, you will get a slightly different plot at the right-hand side. However, the poles will remain exactly at the same positions where they are. So it is still telling you the same thing is going on, that as you start making the network plastic, different symphonies start playing at different points. So that might be interesting in a biological setting. But okay, so here what we can do is just to summarize this point, what we notice is that there seems to be at the edge of stability, complexity seems to have a high value, and as I said, one of the reasons is that that's the point where it relates to this notion of synchrony, and at some point there's an information overload and it breaks down. Okay, this is all right. Let this be now. I more or less described it in other words. Let's go to a bit to the applications, what we can do with it. So just I'm towards the end anyway, so don't worry. So okay, whatever I did was for those small networks, but now what about the original DSI network of Hagman? This is Hagman's data, 998 nodes. It's, as you see, the structural connectivity matrix is quite sparse. So the diagonal itself is not empty, but you don't see it from that far. These are short distance connections and a few long distance connections, and these are all weighted. For sure, at this level I cannot give you a analytic expression, but we can still do numerics. So what we do is we take Hagman's data as it is, with all the weights as they are, and in the only thing is for the A matrix, the adjacency matrix in the linear dynamical system, you can put a overall coupling, a scalar, which you tune over here. So just note that I'm still doing this stuff for the linear dynamics. I've not yet gone to the nonlinear thing. So the results you see are still qualitative, but what you see is all right. So one thing is numerically you can do that, you get something, but what do you compare it with? So over here, as a comparison, what I did was take this network and just randomize it, which means take all the edges as they are with the same weights and just redistribute them randomly. So and compute the same measure for that. So if you do that, what you see is a cross coupling. The red one is the actual data, the actual network, and the blue one is the values of phi. This is all within the stationary regime for the randomized network. So what you see is that, okay, of course, when the coupling is really low, it means this is not very different from a random network, but that's just because very little is connected anyway. As the coupling increases, you see that the structural network with some pattern seems to be doing better as a measure of complexity or as a measure of information integration than a randomized network. So that matters. you might say that it doesn't look like a great difference, but really to say what is a great difference or not, you need something to calibrate it. So coming back to Tony's question, now what you can ask is, okay, what do I have here? Here I just, actually, both David's question and Tony's question, first thing what you could do is, instead of linear dynamics, if you do this for nonlinear, how much more complicated it gets. And then, instead of just considering a neural network, if you now started having, like in the first graph I showed you, each node, instead of just being a neuronal thing, if within that you start coupling it to like a protein expression network or something, one way to do that would be, for example, in a neuronal model, you have an external current. Those external currents, usually we just take them as some external noisy currents, but you could also take them, consider synaptic currents added to the neuronal model. And those synaptic currents might be based on on dynamics that is coming from, say, the CAM kinase pathway or something else like that. So this is what people have done when they looked at treating synapses as switches and all. So you could indeed make the level of modeling more complicated by adding another layer of these interactions, and then ask how much does the complexity at a given value qualitatively increase? Is it true that biological systems really are more complex than the ones that we just simulate as RC circuits on a computer, believing them to be neurons? And if it is true, how much? So that's the kind of thing you could ask over here. However, even though that might be a complicated question in itself because of understanding the dynamics, what you can still do is, as a calibrative comparison, you could start using this measure more for practical surgical data, for practical clinical data. You could start comparing networks of healthy control groups and patients, either which are structurally deficit or which might be dynamically disabled or having problems because of some deficit dynamics. And then you could ask, as a whole, can we quantify the differences between these networks in terms of this information complexity measure? Or another way of looking at it is, instead of talking about the whole network and comparing them, you could even then really just treat them as different parts and try to say how much is the information flow. But you have to be a little more careful in doing the parts business, but at least for the whole business, one thing might be to people in the consciousness field are really just trying to hope that some information measure explains consciousness. A more practical way would be to actually use this for time series data and try to make qualitative comparisons and try to understand if really information processing in certain regions of the brain, when disabled or enabled, or certain kinds of symphonies that might be enabled or disabled, are causing some kind of disorder. So there would be a more practical way to look at these measures, and that is what we are sort of going next for. But of course, to do that, we would first have to now combine it. So we already have been doing modeling with relatively more biophysical dynamics. So the next step would be to include that kind of modeling into these measures and really compare with the data. So that's what we hope to do. And just this last point, all the stuff I said, you could either use it for neuronal models, or you could even use it for decision-making models from monkey data and others, which are very similar to these population models, or genetic switches. But of course, when you do it for different systems, you have to ask what it means. You can't just compare two different systems like that. I mean, you might get some value in bits, but you need something to calibrate it. But nonetheless, these measures are a universal way of addressing these questions of how parts put together work into dynamical complex interactions. And just to thank all the people, the ones who pay us, the ones who share data, the ones who work with us. Thank you.
Thank you, Xerxes. Questions? Where's our second microphone, Tony? Did you hide it somewhere? Yeah.
Hi. I've collected a few as you were going through. I didn't want to distract your kind of flow too much. Going back to some of your early slides when you were showing the activity in the brain as a function of the noise, so quite early on.
Okay.
The reason I asked what the noise units were was I wondered if you had gone down to much smaller units, maybe down to 0 for noise, for instance, or because this is one order of magnitude, and I guess does the behavior change again qualitatively if you go down to very small or 0 noise?
No, we didn't go to 0, but compared to 0.01, in fact, in Gustavo Deco's paper, if I remember correctly, they tried 0.001 or something. There was not much difference. Lower than that,
yeah,
there was not much.
So
difference.
that's really the
Yes.
kind of pattern. Okay.
Right.
Also, a bit later on, I think you showed some typical firing rates, and these were up to, I don't know, 60, 80, 100, 120 hertz. Is that right?
Well, of course.
These are high rates.
Yeah, but those numbers are chosen.
Okay, because of the way you've set up the dynamics.
So what they do is in order to, so these numbers will change according to the couplings. So what people do is in spiking neuron models, they look at what the firing level is like, what they know they should expect from physiology, and they tune couplings so that that fits, and those things are just tuned.
Okay, and I mean, going on from that, obviously this all, I should say that obviously I think information theory for this type of work is exactly the right tool for the job, and you're doing the right kind of approach here to try and grasp a very complex system using information theory as a kind of good starting point is to have to do this. Have you considered how much information is discarded by treating these as populations with rate codes compared to the information that would be stored in the population with timing codes?
So would it be possible? You mean to say that instead of just considering rate models,
Yeah.
of course depends on dynamics and choosing. If I rather consider maybe spiking models or something.
There is some evidence, I think, that there's a lot more information in a parallel rate code, timing code,
Right.
than there would be in a rate code from that population.
Right.
So,
Of course, over here the choices were made according to the data that I can match to.
sure, yes.
So let's say if I want to do that, then I don't need to really look at DTI data of the brain. I might just look at some specific macaque circuits or something that you actually have recordings of individual.
It's a difficult thing to do. I'm not saying you should
Yeah.
have done it. It's a very difficult
No,
thing
no,
to
I
do.
know.
But if we're working with information
Yeah.
content, then it's worth considering. And obviously also the decision to choose it as multivariate normal and such. These are all
Yeah.
all simplifying assumptions that you have to make to be able to
Right.
make progress. But it's worth bearing in mind, as I think some of the other people said, that
Right.
nonlinearity and such will have an impact on this. But,
Right. I just from what I can say, speculate, I would guess that there is it would do something to it in the sense that, just as I mentioned, that instead of just talking about neurons, if you added more structure, you would expect that the values of phi go up. Similarly, considering models that actually have all this timing information, I would think that there is
there's
something more, but
yeah.
I don't have a yes or no answer.
And it's very difficult to even do it at this level. If you then try to take that level as well, that's another, couple of orders of magnitude, probably of complexity.
So let's say it depends what you want to do. If you want to do things analytically, things get difficult, but numerically, it's all doable. You just need a supercomputer or cluster or something.
Yeah.
It's doable. But
Okay, and one other question. When you showed the equation with the Kullback-Leber divergence, was that... Yep. Are you showing the Kullback-Leber divergence between the joint and the product of marginals there? Is that what that is?
yeah, so this is joint in the sense that this is the distribution for the multivariate
Yes,
vector.
the joint distribution.
Yeah.
And that's the product of
And
the
that's the
marginals.
product of the
So that's the mutual information.
So the thing is that, so first of all, just to be a little more careful about this, when these marginals are not chosen by in the usual way where you just integrate off, because then it would mean that you're still having the network as a whole. These are really independent paths.
So it's not really exactly
No.
the mutual information.
No,
Okay.
and the other reason it's not is because normally when people are talking about Kullback-Leibniz, they are not talking in terms of these conditional variables. If you just talk about the variables themselves, then they are the identities to mutual information. When it's conditional, the algebra is a little bit different.
Yep.
It's not always conditional. However, that said, when these distributions are maximum entropy distributions, they happen to be mutual information.
Yep.
Not mutual itself, but the whole versus parts of the mutual. But for any other distribution, it's not generally true.
Yep.
When you have conditionals here.
Yep. Okay.
Yeah.
Thanks. And I think one more thing I would say is that the paper I mentioned to you earlier, the Roderick Little one, I have this with me. I'll show this. I think this will be of great interest to you because it's information theory. It's how to partition large complex networks, and so, but from a more psychological perspective rather than a mechanistic one. So I'll show you afterwards.
Okay. Great. Thank you. Other questions? Okay, they're in the back. So Xerxes, while I'm walking to Stefan, Henry Kennedy told us about these exponential scaling laws about cortical connectivity.
Right.
Right. So if that's true, you would expect that if you now look at this Heckman data set, that you would, if you would impose these kinds of scaling laws of connectivity, you would find, let's say, interesting points or points of high phi. Is that, would you expect that? Have you looked into that?
Not looked into it, but this is something we are considering for the Connectome project anyway. So if indeed we are missing 80% of the gray matter, one thing would be to just take the statistics that Henry Kennedy is proposing and impose not the exact connectivity, but the statistics of that local connectivity, because it's mostly local, he says, and then try to see what happens, one thing at the level of the modeling itself, the phenomenology, if it better explains just the fMRI data, and then for sure at this stage to see what it says in terms of complexity, whether those things are just a little bit or whether that contributes significantly to it. It is something that would be useful to consider. Yeah.
I wonder if you ever considered to use phi as a design principle. I don't even know if it's possible, but let's say you have a set of nodes and you want to, you are allowed to add one connection, and you want to add the one connection that will improve the most the information created by the network. Can we even think about this?
You can think about it in a slightly different way. I mean, just as a fun application, what you can think about is, you can, it's related to this, but I'm not necessarily, I don't know if that's how you meant it. You can think about these questions as, what makes a team a good team? When is a player adding to a team, or is he just screwing things up? So you have different nodes, they have well-defined identities, some statistics, and you put them together. And then you have a new player. Is he enhancing the team in terms of how much it synchronizes and integrates or not? You can do those kind of things, and you can do some sociological experiments on that. But more from engineering designs, a practical thing to do would be to now not just look at networks as they are, but to look at networks in terms of, let's say, instead of just talking about a network, you talk about really a control system, like a LQR controller or something, where you have the dynamical system, then you have the feedback loops. You actually give an identity to all of them. And then you start asking if these measures pick up, do better when the system is under some kind of control or not. Perhaps they would, because after all, when you're controlling, you can filter noise, you can enhance efficiency, you can do all sorts of things. So that might be a more direct thing to test.
All right, great. Xerxes, thank you. As you can see, Tony is hungry. He has to rush off to lunch. Anna, are there any announcements? No. Okay. Get on your projects. Okay? All right. Lunchtime. See you later. Thank you all very much.